DNA, RNA. These two are the most important molecules in living organisms. The entire molecular biology revolves around the DNA and RNA. In the next few classes, let us try to understand the structure, architecture, and functions of the DNA, RNA. DNA is deoxyribonucleic acid, DNA. RNA, ribonucleic acid, RNA. DNA is a polymer of deoxyribonucleotides. Poly means many. It is made up of many units of deoxyribonucleotides. Whereas RNA is a polymer of ribonucleotides. This is a deoxyribonucleotides, this is ribonucleotides. In DNA and RNA, basic units are nucleotides. This is a very important point. What are the units of the RNA, DNA? Nucleotides. What are the units of the carbohydrates? What are the units? Glucose or monosaccharides. What are the units of proteins? Amino acids. Similarly, units of the DNA are nucleotides. Here, deoxyribonucleotides, Suppose one deoxyribonucleotide is ATP. In ribonucleotides, it is ATP. So, this ATP may be present in DNA, RNA. In order to differentiate, we use prefix D. DATP means this ATP is present in DNA. Suppose if you simply indicate ATP, it indicates it is present in RNA or sometimes DNA if you don't mention it. So, in order to differentiate the nucleotides present in RNA, DNA, we use D for the nucleotides present in DNA. Nucleotides are complex molecules consists of three components, three units, three different types of units. Complex molecules, three components. One, a nucleotide consists of a nitrogen base, a sugar, a phosphate or phosphoric acid. There are three components. Structurally, functionally, these are different. Nitrogen base, sugar, phosphate. Nitrogen bases are heterocyclic, means they are cyclic structures, but within the cycle there will be different types of atoms, heterocyclic. These nitrogen bases are again two types, purines, pyrimidines. Purines are bicyclic double ring structures, whereas pyrimidines are monocyclic or single ring structures. Later I will explain the structures of these molecules in detail, next class. Then purines are adenine, guanine. Pyrimidines are three types, cytosine, thymine, uracil. In purines, adenine, guanine. Pyrimidines, cytosine, thymine, uracil. To simplify, adenine is always indicated by A, guanine by G, cytosine by C, thymine by T, uracil by E. just like ATGC or CGTA or GU, like that. A means adenine, G means guanine, C means cytosine, so on and so forth. Then sugar present in RNA DNA is always a pentose sugar consisting of 5 carbon atoms. Pentose sugar. These pentose sugars are two types, ribose, deoxyribose. The ribose sugar formula is C5H10O5, C5H10O5, strictly following the typical carbohydrate structure C1H2O1, and deoxyribose sugar C5H10O4, one oxygen less here. Because of less of one oxygen, it is known as deoxyribose sugar, and because of this, it is known as deoxyribose nucleic acid, ribonucleic acid, one oxygen less. Then this ribose is always present in RNA. Deoxyribose sugar is present in DNA. Now, nitrogen base, NB means nitrogen base plus sugar, nucleoside. Nitrogen base plus sugar plus phosphate is a nucleotide. See the different spelling and also the words. It is nucleoside, it is nucleotide. Here only two nitrogen base sugar, whereas in nucleotide, nitrogen base sugar phosphate. Then these nucleosides, again, ribonucleosides, deoxyribonucleosides. The ribonucleosides are containing adenine, guanine, cytosine, uracil. Similarly, deoxyribonucleosides containing adenine, guanine, cytosine, thymine, but not uracil. Thymine is present only in DNA, but not in RNA. Similarly, uracil is present only in RNA, but not in DNA. This is again very, very, very important. Many times it is asked in the examinations, different types of exam. Nucleotides, ribonucleotides, deoxyribonucleotides. So this is the basic composition of the nucleotides. The nucleotides in turn the units of the DNA, RNA. And now let us consider in the next class or in continuation of this class the structures of these nitrogen bases, sugars, phosphates, how they are linked to each other form the nucleotides. Still in another class, we will see how these nucleotides polymerize to form a definite structure known as DNA. Okay, this is. Now let us try to understand the chemical structure of the components of nucleosides, nucleotides. The three components are nitrogen bases, sugars, phosphate. Now, when we take the nitrogen bases, purines, pyrimidines. Purines are adenine, guanine. These two are bicyclic with two ring structures. Total 9 atoms. At the 5th position, at the 7th position, at 9th position, nitrogen. Remaining positions, carbon. This 9th position, nitrogen, is important for us. The difference between adenine, guanine Adenine and guanine is here you see. In adenine, at the second position, amino group, here oxygen. In guanine, at the sixth position, amino group, but there is no such amino group. Then pyrimidines or thiamine, cytosine, uracil, single ring structures. These are with 6 atoms. The rings are made up of with 6 atoms. First position, third position, nitrogens, remaining positions, carbon. Now, if you see carefully, thiamine and uracil. So this thiamine differs from the uracil in having methyl group at the fifth position. Therefore, it is known as 5-methyl uracil. The differences between cytosine and thiamine uracil are here. In cytosine, there is an amino group. So, thiamine, cytosine, uracil. Then this is a phosphate hydroxyls. So, these are 3 hydroxyls functional groups which can actively participate in chemical reactions. Then coming to the sugars, deoxyribose sugar, ribose. These two are pentose sugars. In fact, these sugars will be straight, but here what happens, they form a ring-like structure, Purines structure. 1, 2, 3, 4, 5. These 4 make a ring-like structure and it will be a boat-like thing. The 4 carbons, 1, 2, 3, 4, 1, 2, 3, 4, they are within the ring, make the ring, but the 5th position carbon is outside the ring. And look at the difference between the ribose and deoxyribose. In ribose at second position, there is hydroxyl group at second position. But whereas in deoxyribose sugar, there is no hydroxyl. Therefore, it is known as 2-deoxyribose. At second position, there is no oxygen, there is less oxygen. So, therefore, this is C5H10O4. This is C5H10O5. So, this is important again for forming the bond. The first carbon is important, the 5th carbon is important, and also the sometimes third carbon. Now, nucleoside. Nucleoside is formed by the 2 components. One is a nitrogen base, another is a sugar. This nitrogen base may be purine or pyrimidine. If it is purine, it may be adenine or guanine. Then sugar may be ribose sugar or deoxyribose sugar. Then whatever it may be, whether it is purine or whether it is adenine or guanine, then a linkage will be formed. That linkage between the nitrogen base and sugar is known as N-glycosidic linkage, N-glycosidic bond. Linkage or bond between the nitrogen base and sugar. And further, this linkage will be formed between the nitrogen of 9th position of the purine and first carbon of the sugar, N9 of purine, C1 of sugar. This is N-glycosidic bond. Suppose if it is adenine, this is called adenosine. if this is guanine, guanosine, this nucleoside will be known by guanosine. Then if the nitrogen base is a pyrimidine, thymine, cytosine or uracil, the same glycosidic bond between the nitrogen base and the sugar, but the difference is this glycosidic bond is formed between the first nitrogen of pyrimidine and the first carbon of sugar, N1 of pyrimidine, C1 of sugar, and glycosidic bond. If this is thymidine, if this is cytosine, then that nucleoside will be known as cytidine. If it is uracil, then uridine. Now coming to nucleotide. A nucleotide consists of three components: a nitrogen base, a sugar, a phosphate. Now let us see here. This is nitrogen base, any one of the five. This is sugar. This nitrogen base, sugar are linked by N-glycosidic bond, as I have explained you here. This is N-glycosidic bond. But this sugar is linked on the other hand through phosphate. So this sugar is linked on one hand with nitrogen base by N-glycosidic bond, other with phosphate by a bond known as ester bond. This is known as ester bond. This is N-glycosidic bond. So this is nucleotide, nitrogen base, sugar, phosphate. In a nucleotide, there are two linkages. One N-glycosidic bond, another is ester bond. But this phosphate is linked to another nucleotide in the chain through a sugar. Here this is also ester bond. So therefore this phosphate, on one hand it has one ester bond, other ester bond. Therefore this is known as phosphodiester bond. On either side two ester bonds, phosphodiester bond. Now here this is, suppose this is sugar. This phosphate is linked to sugar with C5, this. But this phosphate is linked to this sugar with C3, this. Phosphate, one hand, sugar with C5, another with C. Therefore, a nucleotide will have two ends, one 5, another 3. Therefore, the DNA chain is also designated as 5', 3'. Generally towards 5 side phosphate, towards 3 side hydroxide. This is nucleotide. So, nitrogen bases, phosphate, sugars, nucleosides, nucleotide, known by different names depending upon the presence of the nitrogen, type of nitrogenous, type of sugar. Suppose if this is adenine, if this is deoxyribose sugar, deoxyadenylate. If this is uracil, if this is ribose, then this will be known as uridenylate. That is, in the next we will see the structure of the DNA with the knowledge of these structures, because in order to understand the DNA structure, RNA structure, we must perfectly, clearly understand the structures, the bonding and other aspects of this.